A common question asked by people around the world is why science has not yet discovered a cure for cancer. Nearly 1 in 5 people will be diagnosed with cancer in their lifetime, which is an incredibly high statistic. Globally, astronomical sums of money have been poured into cancer drug development. And yet, despite this, cancer remains one of the most fatal and persistent killers of humanity, ranking as the second leading cause of death in humans. So, why is science struggling to combat it? Well, I was wondering this myself, and today I'm going to break down that mystery. And don't worry, by the end, I'll cover the promising trials and upcoming therapies that have come from these years of cancer research. Just to make sure we're all on the same page, I'm going to define cancer quickly, and then we'll go into some types of cancer and how it works. Cancer is a group of diseases characterized by uncontrolled abnormal cell division. Cell division is just the process through which a cell divides itself into two separate daughter cells, and this is how things grow bigger and how bodies heal. Although cancer is grouped under one term, there are many different forms of cancer. For instance, melanoma, a type of skin cancer, originates from pigment-producing skin cells that have changed due to sun damage. In contrast, breast cancer develops from glandular cells. They develop in different ways, in different areas, and attack the body in different ways. So what makes them both cancer is their shared underlying disease process, which is a complete breakdown in the rules governing cell growth and death. To better understand how cancer develops, I'll first define two terms. First, DNA, which you may or may not know, is the master instruction manual inside every cell, containing the recipes for all proteins. Second, mutations, which are random, permanent spelling errors or changes in the DNA manual, caused by either a copying mistake or environmental damage such as radiation and toxic chemicals. In a healthy cell, certain genes control accelerator and brake pedals. They can press the accelerator pedal to promote cell division, or press the brakes to stop it. Mutation jams the accelerator pedal on and rips off the brake pedal, putting a cell into a state of endless cell division. Now, when I was researching this topic, I was thinking to myself, well, a lot of diseases work through mutations, so why are things like bacteria or viral infections easily cured by medicine, but cancer isn't, if they're both a matter of mutation? I discovered that the answer lies in a concept called selective toxicity. When treating a bacterial infection, antibiotics act as highly specialized wrenches that target structures unique to the bacteria, such as bacteria cell walls. Because cancer is formed by our own human cells and not an infectious bacterium, this antibiotic wrench has nothing to grab onto. What makes cancer cells so unique is that the cancer cells are not foreign invaders, they're the body's own normal cells. They use the exact same machinery, enzymes, and pathways as healthy cells. There's no unique bacterial gear to jam. This biological similarity creates a problem for therapies. If we make a drug that targets the cancerous cell, it's highly prone to targeting healthy cells, since they have the same appearance. And if we make a drug that targets rapidly dividing cells, it can still target healthy cells, since bone marrow or gut lining cells also rapidly divide. So now you're probably starting to see part of the problem. But that is not the only issue. To make matters worse, tumors are highly dynamic and genetically unstable. This basically just means that tumors are super hard to diagnose. A single tumor is not a uniform mass of identical cells. It's a branching evolutionary tree of genetically distinct subclones. This leads to a big blind spot. Say we use single-site tissue biopsy to find the genetic makeup of a small part of that tumor in order to create a specialized medication for it. Well, since a tumor's genetic makeup is super vast, that sample taken will probably make up less than a quarter of that tumor, meaning that specialized medication will only work on a tiny part of that tumor, meaning that the rest of the tumor will easily be able to replenish. This large amount of diversity creates persistent subpopulations, the most notable being cancer stem cells, or CSC for short. CSC are very notable for being sneaky. A lot of current therapies target rapidly dividing cells, but this type of therapy acts like a lawnmower. Although it cuts down the tall weeds, it can't pull out the roots, meaning that these weeds are able to grow back. In this analogy, the weeds are CSCs. These cells remain dormant by resting in the G0 to G1 phase of the cell cycle. This resting state is regulated by highly conserved signaling pathways, which keep those cells alive and highly resistant to therapy. What makes this so difficult is that CSCs, due to those signaling pathways, can detect changes in their microenvironment and become dormant for periods of time in response to threats. This is different than most other types of cells, which will self-destruct when sensing infection, bacteria, or DNA damage in order to protect the rest of the body. This self-destruction is actually very important in order to maintain a healthy body and replace old cells. So now we get into how current medications like conventional chemotherapy function against these difficult circumstances. Standard chemotherapy agents are designed to stop cell division or trigger cell death in rapidly multiplying cells. Although this can be effective against cancerous cells, this is why people who undergo chemotherapy lose their hair, develop sensitive stomachs, and can potentially become infertile. Your hair cells, digestive tract cells, and reproductive system cells are good examples of cells that naturally rapidly divide in your body, and chemotherapy just wipes all good or bad rapidly dividing cells. Chemotherapy helps to suppress and shrink primary tumors, and undoubtedly is an extremely helpful treatment. The reason chemotherapy doesn't have a 100% success rate is because, as discussed a little bit earlier, it can miss dormant CSCs. Along with those cells, it is possible for cancerous mutated cells to possess drug resistant mutations. Since mutations are totally random, cancerous cells divide so rapidly and diversify so much that it's possible that there are cancerous cells in a tumor that are unaffected by chemotherapy just randomly due to the way they've mutated and evolved. If this occurs, these resistant cells, no matter how few, are the only survivors while the rest of the cancerous population has been eliminated by the chemotherapy drug, meaning that these small population of cells will make up the new dividing cancerous cells, leading them all to carry on this mutation that makes them chemotherapy resistant. This process is functionally identical to the way that bacteria develops resistance to antibiotics. So, to sum up all of that information, we basically have two main reasons that cancer is so hard to treat. First, cancer is such a wide umbrella of harmful cells that there is not one type of cell for a medication to target. When cancerous cells rapidly divide enough to create a tumor, tons of these cells become mutated due to this rapid unchecked division. Mutations continue to occur and create a ton of unique cells with differing roles and appearances. When there are so many subpopulations of cells that look and act different, a medication would need to be developed for every type of mutated cell within a tumor, which is basically impossible to do because these cells are constantly mutating and therefore constantly changing. Secondly, cancerous cells are mutated versions of our regular bodily cells, meaning that a medication created to target these certain types of cells that become cancerous would just kill all populations of those types of cells, healthy or cancerous, because they share the same form. And in many cases, the healthy cells being killed would create serious issues within the person's bodily functions, leading to possibly death. Cancer mutating from our own bodily cells also means that it knows how to manipulate our body's pathways without getting caught. Typical diseases that arise from bacteria are able to be combated by our body because our immune system cells recognize that these bacteria cells are foreign and not something that's usually in our body. But with cancerous cells, they're made from naturally occurring cells in our body, so our usual defense systems do not detect them as foreign and therefore don't fight them. Given those hurdles, what is modern scientific research currently focusing on? There are many therapies in the works, but I'm going to be discussing two very prominent and promising ones. First, we have the antibody drug conjugates, or ADCs. ADCs are meant to target cell markers instead of cell types. You can think of cell markers as a molecule inside or outside of a cell. If a cell becomes cancerous, it has a higher amount of tumor markers compared to regular cells. So these ADCs are meant to target these markers, specifically the tumor markers, and then bind to the specific molecule on the cell and inject toxins directly into the cell. This solves the selectivity problem discussed earlier, where medications target a cell type and in the process hurt healthy cells as well. If ADCs can target markers instead of cell types, we can bypass that problem, although it's much harder to create an antibody that seeks out markers, which is why these antibodies are still in development. However, more than 15 ADCs have been approved globally to target certain cancers such as breast, lung, and bladder, and over 40 ADC candidates are currently in phase 3 of clinical trials. The second prevalent therapy is CAR T cell therapy. T cells are basically the immune system's highly trained soldiers. Normally, cancer cells evade T cells by putting T cells to sleep through binding a protein to their receptors. In CAR T therapy, scientists extract your body's natural T cells, genetically upgrade them with custom receptors to recognize the cancer cells and avoid being put to sleep by them, multiply those cells, and return them into the patient's body. This overcomes the tumor's ability to evade the immune CAR T cell products, which mainly focus on blood cancers such as leukemia. This therapy is still under production for treating solid tumors and expanding to treat other types of cancer. Due to the sheer genetic diversity and constant mutation of tumor clones, a single universal cure is highly unlikely. However, by combining ADCs to destroy the bulk of the tumor and T cell therapies to seek out the hidden survivors, future medication aims to transform cancer into a more manageable chronic condition. And the best scientists in the world are on the task. So that is it for my short form answer to why cancer is so hard to cure. I hope you enjoyed and learned something.